And there's remnants of genetic information, remnants of ribosomal RNA from all 4 of the organisms present. And this is why the biologists get so hot under the collar and get so mad and they yell and they change the classification every year. Okay, so again, just so we understand why this group is so hard to classify. So are we going to classify them? No. All we're going to do is talk about, can they move? What is the structure they use for motility? And the other thing we'll talk about is their lifestyle. How do they make a living? Are they photoautotrophs or chemoheterotrophs? Are they free living or are they parasitic? That's just what we're going to stick with, okay? All right. So as we mentioned, we're going to look at 4 groups of these protists. Most of them are unicellular eukaryotes. Okay? And again, we're just going to break them down according to whether they're motile. If they're motile, what structures they use. So the 4 groups we'll look at are the flagellated protists, the ciliated protists, the protists that are amoeba, that use pseudopodia, false feet for movement. And then, as we said, next Tuesday, we'll take a look at the protists that aren't motile. They're called apicomplexan. So we're just going to look at a few examples of each of these guys. We're going to start out first looking at the flagellates. Okay, the flagellates. So here's our flagellates. And one of the things we want to remember when we talk about these flagellates is that the eukaryotic flagellum is very different in structure from a bacterial flagellum. And what we'll do is we'll use our model of one of our flagellated protists. This is a model of the organism Euglena. So here is its lovely flagellum. Now, in contrast to bacterial flagella, eukaryotic flagella are covered in cytoplasmic membrane. You guys remember that bacterial flagella, they're just naked protein. Okay, they aren't covered by cytoplasmic membrane. So eukaryotic flagella are covered by cytoplasmic membrane. The eukaryotic flagellum is stabilized by these long protein tubes called microtubules. And those of you that have had A&P, this probably sounds familiar. Okay, yeah, this 9+2 arrangement of microtubules. So what that means is if we were to take a cross-section through the eukaryotic flagellum and put it on a microscope slide and look at that cross-section, we would see, okay, so this would be a cross-section of the flagellum. What we would see is 9 pairs of microtubules around the periphery. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so that's the 9. And then there's 2 single microtubules in the middle. Okay, so that's the classic 9+2 arrangement of microtubules in eukaryotic flagella. Okay, very different from bacteria. Yeah?
You said the flagellum's covered with the
cytoplasmic membrane, yeah. So if we could imagine this flagellum, it's actually covered in an extension of the cytoplasmic membrane. It looks naked here, that's just because it's a model, right? Okay. And another difference between bacterial flagella and eukaryotic flagella. You guys remember in bacterial flagella, they rotate like a boat propeller, right? Mm-hmm. Okay. A eukaryotic flagella, they whip back and forth. That's how they create the propulsive force. Okay, they whip back and forth. They don't rotate like a propeller. Okay. All right. So if we look at protists that have flagella for motility, we call them flagellates, just as a general term. And we're going to look at a few different examples. Okay. The first one we'll take a look at is a free-living flagellate, Euglena. And this is our model of Euglena right here. Now, in your second handout, you guys, it has a few little handwritten notes on front. The second page, we Xerox the keys that go with these models. Okay. And in addition, the keys have many more parts of the models labeled, so we don't want you to fret about all the parts. What we tried to do is, for example, here's the little Euglena model down here. We put Euglena no, and then we just labeled the parts we want you to know. And these models do appear on lab practicals. Okay, so we're going to go over them together to make sure you know the names of the parts and their function. Okay, so first of all, on the lab practical, if this model was at a station, we'd ask you to provide the genus name. So what's the genus name? Euglena. There you go. You're spot on, you guys. Okay, so let's take a look at this guy. Identify the structure at the tip of the pointer. Flagellum. The function? Motility. Dynamite. And just so you all know, I always thought when I first was learning about these cute little guys that the flagellum extended like this and it would whip back and forth like that. But it turns out it actually bends back over the so-called body of the euglena, and it whips back and forth and pulls the euglena along like so. Yeah, isn't that cool? Oh, they're so fun. They're so cute. Okay, now the reason these guys are green, why are these guys green? Chloroplast, right? Remember they're eukaryotes. This is the nucleus. Okay, they're eukaryotes, and eukaryotes have their chlorophyll A and membrane-bound organelles called chloroplasts. Okay, how did the chloroplast evolve? Where did the chloroplast come from?
Cyanobacteria.
From cyanobacteria. Dynamite, you guys. Excellent, excellent. So that's where chloroplast evolved from. Now, to make this even messier, you guys are not going to like this. I was always so confused because it turns out euglena, if there's no light available, it can act as a chemoheterotrope, okay? But if light is available, it carries out photosynthesis, right? And it gets classified with some of the parasitic protozoa. I'm like, I am so confused what's going on here. So just so you know why this is so confusing, it turns out euglena acquired its chloroplast by secondary endosymbiosis. It started out originally as a chemoheterotrope. It was a hunter itself. It hunted algae. Some of the algae took up housekeeping inside the cytoplasm. So it acquired the chloroplast later in evolution. And that's why it's a mixture of like an animal-like protist and a plant-like protist. Okay, just so we understand why this guy is so challenging. Okay, so again, you identified the structure as a chloroplast. What's the function? Photosynthesis. If it has, exactly, if it has chlorophyll A, which type of photosynthesis? Oxygenic photosynthesis. Dynamite, you guys. Excellent. And again, just to make sure, what's the structure at the tip of the pointer? Nucleus, right? And this little dark spot is called the nucleolus. This is where ribosomal RNA and ribosomes are assembled. Great. Okay, this is very cool, you guys. This little red dot is called the eye spot or stigma. It's part of a light detection or photo detection system. It makes sense if this little guy is photosynthetic and it's living in, say, a pond. It has to have some light detection system that will help guide it to the place in the little pond where the maximum amount of light will be located. Okay, so we call that phototaxis. Remember chemotaxis, movement along a chemical gradient? These little guys exhibit phototaxis, movement along a light gradient. Very cool. Okay, almost finished, you guys. This little thing that looks like a marble, this is called a contractile vacuole. Its function is to pump extra water out of the cell so it won't undergo osmotic lysis. Okay, it won't undergo osmotic lysis. So the contractile vacuoles, the function is to pump water out of the cell to prevent osmotic lysis. Okay, and let's just double check our list, you guys. I think I forgot one thing. Pellicle. Okay, it turns out euglena doesn't have a true cell wall, okay, but it has a protein reinforced cytoplasmic membrane we're going to call a pellicle. Okay, so if we tagged like this side of the euglena and said identify the structure, we had an arrow pointing here, what we're going for is what? Pellicle. Pellicle, right, you got it. Good job, you guys. Very good. Okay, so our cute little euglena. Yeah.
I thought the head was called the pellicle.
Oh, the pellicle is the reinforced cytoplasmic membrane. So it's the whole thing. On the model, or on the key, you're right. It maybe points here, but it's the whole protein reinforced cytoplasmic membrane is the pellicle. Yeah, good point. Thanks, Al. Thanks, Alex. Okay. So that's an example of our free-living flagellate. Now, there are a few parasitic flagellates that we want you guys to learn. And what I'm going to do is I'm going to move this over to the sideboard so we can expand our description of them a little bit. So, okay. So